Welcome to this explainer. Today we're unpacking Chapter 5, all about cellular processes. But we're actually going to shift our perspective a bit here. Instead of just picturing a cell as some static, boring blob of jelly, I want you to imagine it as a hyper-intelligent, absolutely bustling metropolis. Seriously, just like a massive city, a single cell has its own telecommunications networks, power grids, and even a highly specialized job market. So let's dive right into this incredible microscopic world.
Now, you might be wondering, how exactly does a microscopic blob know exactly what to do? It's a fascinating question, right? How does this tiny, seemingly simple little structure manage incredibly complex survival tasks in real time? How does it know when to grow, when to use energy, or when it's time to divide? Well, the secret lies in these highly coordinated systems running perfectly in the background. Systems that honestly rival our most advanced human engineering.
So here is our roadmap for the city today. We'll be covering 4 key areas. First, how cells communicate. Second, the cell's power grid. Third, glycolysis and energy production. And finally, stem cells and differentiation. Let's get into it.
All right, Section 1. How cells communicate. Think of this as the cell's telecommunications network. Just like any city relies on phone lines, Wi-Fi, and fiber optics, cells act like a massive telecom network. They are constantly receiving, interpreting, and responding to signals from their environment. To really get this, let's define 2 key terms. Think of a ligand as a highly specific chemical messenger. Basically, it's a text message sent out into the cellular environment. But, a message needs a receiver, and that is the receptor. This is a specific protein sitting on the cell surface, or even right inside the cytoplasm, that acts just like a receiving antenna. Now, a cell can only respond to that chemical messenger if it actually has the exact matching receptor. If there's no matching antenna, the message is completely ignored.
Depending on how far apart the sender and the recipient are, this communication mostly happens in 3 different ways. First up is paracrine signaling. This is your short-distance communication. Think of it like instantly passing a note directly to your neighbor, which makes sense why it's commonly used by those rapid-fire neurons in your brain. Second, we have autocrine signaling. This is where a cell secretes a ligand that binds right back to its own receptors. It's essentially a cell talking to itself. And finally, there's endocrine signaling. This is the long-distance broadcast. A ligand, like a hormone for example, is released straight into the bloodstream to travel far and wide across the entire body to find its target.
But take a look at what happens when these communication lines are exploited. In normal autocrine signaling, cells rely on highly regulated external signals to know exactly when to grow. Cancer cells, however, act like rogue citizens. They completely bypass the city's zoning laws by actually synthesizing their own growth factors, while already possessing the specific receptors for them. So they basically create this endless feedback loop, just telling themselves to grow and divide uncontrollably. It's a brilliant, but obviously devastating hijack of that normal telecom network.
Moving on to Section 2, the cell's power grid, also known as metabolism.
Okay, so every city absolutely needs a power grid, right? And for the cell, that's metabolism, the vital everyday process of extracting and using the free energy required for life. Now, metabolism is a two-way street. It's constantly balancing the building up and the breaking down of materials. On one side, you have anabolic pathways. These are your construction crews. They build larger, complex molecules from smaller ones, like synthesizing proteins. And because they are actively building, they are endergonic, meaning they consume energy to do the work. On the flip side, we have catabolic pathways. These act like the demolition crew. They break down those larger molecules into simpler ones. These reactions are exergonic, meaning they release energy, primarily in the highly useful form of ATP.
To put that into perspective, in most animal tissues, the primary metabolic fuel, think of it as the raw coal being fed into the city's furnaces, is glucose. Glucose filters through these incredibly complex metabolic pathways, eventually breaking down into pyruvate through a process called glycolysis. From there, depending on whether oxygen is around or not, it either enters the citric acid cycle for a massive energy payout, or it diverts into lactate. It's really this beautifully intricate highway system of energy conversion.
Which brings us to Section 3, glycolysis and energy production. This is all about keeping the lights on.
Let's zoom in on that very first step of the metabolic highway, glycolysis. This all goes down right in the cytosol. It starts when an enzyme called hexokinase phosphorylates a 6-carbon glucose molecule. And here's a really cool detail. This step is irreversible and is allosterically inhibited by its own product. That means the cell actually has a built-in sensor to stop itself from overproducing. So the glucose is then methodically broken down into pyruvate. Now, if the cell is operating under anaerobic conditions, meaning it's lacking oxygen, it undergoes fermentation. This is a two-step process. First, pyruvate is decarboxylated into acetaldehyde, which releases carbon dioxide, and then it's reduced into ethanol.
So the absolutely crucial takeaway here is this number, 2 ATP. That is the net energy payoff produced per single molecule of glucose during this fermentation process. Now sure, aerobic respiration yields way more energy, but this quick 2 ATP payout, that's the emergency backup generator that keeps basic cellular functions running when oxygen is scarce.
Finally, we reach Section 4, Stem Cells and Differentiation. Welcome to the cellular job market.
A city isn't just made of concrete buildings and electricity, right? It relies on specialized workers. Well, inside the body, unspecialized stem cells exist in this really strict hierarchy based on their potency, which is just a fancy word for their ability to differentiate into other cell types. At the very top, you have totipotent cells, like a newly formed zygote. These have the absolute highest potential. They can literally become any cell in the human body, plus extra embryonic tissues like the placenta. One step down from that are pluripotent cells. These can form almost any tissue from the 3 germ layers, but they can't form the placenta. And finally, we have multipotent cells. These have a specialized, much more limited range. A great example would be hematopoietic stem cells, which only differentiate into specific blood cells, like red blood cells and platelets. So as cells step down this hierarchy, they basically trade their infinite potential for a highly specialized skill set.
Now, for a long time, accessing those powerful pluripotent cells meant relying strictly on embryonic stem cells. But then, in 2006, scientist Shinya Yamanaka changed absolutely everything. He developed what's called induced pluripotent stem cell, or iPSC technology. He basically figured out how to take a fully specialized adult tissue cell and retrain it. He induced pluripotency using specific transcription factors. And guys, this is a total game changer. It completely bypasses the need for embryos. And even better, because you can make patient-matched pluripotent cells, it entirely avoids the risk of transplant rejection by the immune system. It is essentially sending a specialized worker all the way back to the ultimate cellular university to learn a completely new trade.
So as we close the gates on our cellular metropolis today, I want to leave you with this thought. We now have a solid grasp on how the cell communicates, how it powers itself, and how it assigns roles to its workers. But with iPSC technology giving us the incredible power to literally reverse cellular time, retraining adult cells to model complex human diseases, what medical breakthroughs are just around the corner? The microscopic city inside us is vast, and honestly, we are just beginning to learn how to read its blueprints. Thank you so much for joining me on this explainer, and keep exploring.